Afternoon session. Thank you for coming to this nice conference. I hope you enjoy it. We have Orlando Guzman, yes, from UAM Iztapalapa. All right, that's you. Thank you very much for accepting this invitation.
So, first of all, I want to thank the organizers for inviting me. I really enjoy to see you. It's a pleasure to meet very good friends. And after a long absence of the conferences because of the pandemics and all of that, it's really good. It's really good to see you. So, this time I will speak about some problems that we've been working with a set of collaborators in the University of Ohio State, Professor Julia Rusina, and in the University of California, Los Angeles, Professor Robin Bruinsma. And the idea is that we're interested in the physics of HIV virion assembly. And part of that problem involves the capture of viral RNA by the proteins that form the capsid. That protein is called GAG. And it's a common name for the proteins that structurally form the capsids of retroviruses. But the story is that HIV is a strange retrovirus. And I'm going to explain why, in what sense it's strange. And because of that strangeness, the task of retrieving the RNA of the virus in the cell and put it into the virions in order to produce infections is harder for this virus. And even today, it's not well known how it does it. And we proposed a model that can explain how this happens. And so I'm going to go over here. Same problem. Okay, it's just slow. Okay, so on the left, there is a cartoon of the life cycle of an HIV virus. The HIV virus is a virus that its genetic information is stored in RNA. And it has a special mechanism to move this information into the DNA of the host. And it stays there for a long time. And eventually, when it is time to create new viruses, it's translated again into RNA. And this RNA is the template that is used to create the proteins that eventually will form a new virus. This part, whoops. This part here, it's kind of funny because this laser is not powerful enough to be noticed. So I'm going to go over here. So in this part, you see that there is some chains of HIV proteins, and these chains are recruited to the membrane. And in the membrane, they line the interior of the membrane, and they help to create some curvature here. And the other thing that they do is that as they are doing this, they capture RNA. This is the genome of the virus that eventually will go away and infect another cell. Now, this part, this assembly of the virus, is the part that we are most interested in. And this is an actual scanning electron micrograph showing or depicting a human cell, a CD4 cell, and these yellow things are the HIV virions. So those guys are the cartoon. Each one of those is one of those cartoons that are there liberated. And each one of them carries 2 copies of a very long, a long RNA chain that is the information that is needed to create more virus in the next sequence of infection. So this is a map of the GAG protein. The capsid is formed by about between 1,000, maybe 3,000 proteins of this kind, which is a kind of long protein of about 500 amino acids or residues. And it's modular. It has several parts, and several parts are in charge of several functions. For instance, MA is associated with the membrane. So its function is to go and find its own way into the membrane. And these proteins are the proteins of the capsid, And this part here, this is the part that forms the nucleocapsid. And this NC is also responsible for locating RNA and taking into the virion. Is the scout that grabs the viral RNA and brings it into the interior of the virion. So in that sense, this is a complicated machine. And we've studied this in other studies. We're using molecular dynamics, atomistic simulation, or coarse-grained simulations to figure out how it works. But one part that really interests us is this. On a percentage larger than 90%, this virion, in the end, carries exactly 2 copies of the RNA genome. Not 3, not 4, just 2. And it lines the interior of the virion. It's associated electrostatically with these proteins. And we would like to know, how is it possible that this thing recognizes in such a large percentage, over 90%, the correct RNA? Because these proteins can also recognize regular human mRNA. And the concentration of mRNA, of the human host, is much larger than that of the viral RNA. By what fraction? Well, some estimates tell us that if you measure the total length of RNA, if you put together all the mRNA of the host and all kind of RNA that is there, it's about 10 to the 5, 10 to the 4 times larger than the total amount of viral RNA in the cell. So there is a conflict. How does the virus find such a tiny amount of RNA correctly in over 90% of the cases? And if you like thermodynamics, you might start to argue that there must be some free energy involved in the association, specific association of the RNA of this virus and these proteins. Maybe there is a special binding energy so large that compensates for this tiny amount. Well, this is a cartoon of what is known about the sequence of RNA that interacts with the protein, the NC domain of the GAG protein. And what has been found is that there aren't too many specific sites where this protein binds to the RNA. There might be 2, 3. It's not 1,000, it's not 100, it's just a few of them. And the interaction energy is not too different from that of binding just regular mRNA. So the thermodynamic advantage of binding this cannot explain thermodynamically that huge specificity of the virus in recognizing its own genome. So that's been a trouble for couple of decades of finding out how is it that the virus is so effective in finding its own viral information. Yes, Nina? This has been done in vitro and in vivo. Yes, and there's also been some studies when you can mess with the mRNA, or with the viral RNA, and disrupt several regions of this. And then you see that if you disrupt these regions, which are not specific for the proteins, then you lose the selectivity. And I'm going to speak about a lot more about this. Doesn't seem to be working. But I can hear you.
Okay, now it's green, but somehow other people can't. Okay, so normally, well, now it's known that most of the viruses, when they infect a cell, they construct viral factories.
Yep.
So it's not that they get into the cytoplasm of the cell and then they have all this whole volume of, I don't know, 60 femtoliters to play with. They actually have everything very highly localized. So if you have the RNA polymerase nearby, copying and the like, then you assemble right in the vicinity.
You're totally correct. One thing that makes the HIV virus stand apart is that it lacks that thing. It doesn't do that.
Okay.
It cannot do it because it doesn't have a gene for that factory.
Neat.
Okay, so what has been observed is that you If you mess with a specific part of this RNA, then you lose selectivity. And that part happens to be a region where the RNA is complementary to itself. So it's a region that promotes dimerization of the RNA chains. And that's the reason why you only have 2, because it dimerizes along this particular sequence. So if you, this is called the dimerization initiation sequence, and there are ways to mutate this structure so that it's sequestered. And if it's sequestered, then cannot dimerize, and then you lose selectivity. Okay, so these are things that must be incorporated in any model that attempts to explain how it happens. And this is a picture that shows this process. So these are fluorescence images of the membrane, and these are RNAs chains that have been labeled with a specific fluorescence marker. So one is red and the other is green. And in time, you can see that they approach and diffuse, and then eventually they bind together, and they are dimerized. And what you observe is that if you have any of these copies, you find a small amount of GAG protein associated with each of these chains. But after they dimerize, the number of GAG proteins that are there increases, and eventually will create a full virion of about 2,000 copies of the GAG protein that are needed in order to create the full sphere of the virion. So that's another clue that we need to put into our model. So what we did is to create a kinetic model. Instead of just saying that the process occurs in equilibrium, we allow for rates of these transformations to occur. And this is a cartoon of some of the processes that are involved. So this is a cartoon that represents somehow the membrane, and this is a cluster of 3 GAG proteins that are attached to some form of RNA. In this case, it's illustrated with just regular mRNA. Maybe they are short chains. And there, in the cytoplasm, there is also some GAG protein that is associated with the small transverse RNA chains. These proteins are highly charged, and they are normally associated with some form of RNA. It could be the viral one, or it could be any other thing. And so, in our equations, what we assume is that this process is constantly occurring. Some proteins are arriving constantly to the membrane, associating with it, and they are constantly detaching from it. So this is a rate process. It's going to be controlled for some kinetic equations. And the equations encode the information about the binding energies and the chemical potential of the GAG proteins in the cytoplasm. So we have access to this information because the experimentalists can measure these quantities as dissociation constants and so on. So if you attempt, or you do the calculation to figure out what is the fraction of viral RNA compared to the competitive RNA that is junk for the virus, this number is kind of small, 10 to the minus 5, 10 to the minus 4. And if you do the calculations, when you take into account some specific binding energy of the order of 2 kBt, you end up with this sort of enhancement of the recognition. So the idea is this, that when you have the possibility of deciding if you're a GAG protein, you can decide to either join the cluster when you take regular mRNA, and it associated some energy, or you could choose the correct viral RNA, and then you gain 2 kBt extra. So it's better if you choose the correct mRNA. There is some ideas that is called kinetic proof reading. That happens when you have one possibility of alternate bindings. And then as the process is repeated in time many times over, you find that there is some advantage of getting the right binding because these bindings are stronger. So if you try many times, those that are correct are enriched with respect to that are not correct. Well, that mechanism, you put it in action here, and what you obtain is that, is that it doesn't allow you to get to the 90% that you want. This mechanism will get you about 5% selectivity, which is not selectivity at all. So it's totally opposite of what you want. And that was the problem that we had when we started analyzing by ourselves these things. Okay, we come to this problem in biological physics from the point of view of statistical physics and non-equilibrium statistical physics. Now, we have to recognize that there are some, let's say, expectations. We cannot have extremely good expectations. We have low expectations because we are humble physicists. And the thing is this. We are not going to make a microscopic model that is so complex that explains everything, but we cannot calculate anything with it. We want to have a simple phenomenological model that will be consistent with what we know about statistical physics. The other thing that we know is that we have a few parameters that we can extract from experiments, and other things we will have to tune. The main thing that we want is, can we make interesting predictions? Can we, first, reproduce the level of selectivity that is observed, over 95% or so? If so, under what conditions? What are the parameters that explain those things? And moreover, can we do experimental suggestions, things to change, mutate in the proteins, in the RNA, so that the experimentalists will test the theory and show that it's wrong or that it's right? So one way to think of these equations and this kinetic model is consider it as a race. There is a race, and there are two horses. One of the horses, one of the channels of production, is to create a virion. We're going to introduce a variable that is the distribution of sizes. We can imagine that as the proteins go into the membrane, there is a cluster that will grow, and the size of a cluster is n. But this is not just a process that occurs in time, because it's more like a random event. It could grow because it has absorbed a new protein, or it can shrink because it has lost one of the proteins. So you can imagine that this is something like a random walk. And there is a different thing, another path, that involves the creation of clusters that do not grow on viral RNA. They grow on the clusters of mRNA. So we're going to explain, or want to explain the selectivity in terms of saying, look, one of those processes occurs faster than the other. And so the selectivity occurs because this is created more rapidly than the one that is not the correct viral RNA that is worthless to the infectious process. This is the idea. So what we need to do is to create this model. So the model has the aspect of a lot of differential equations. We model the rate of change of the distribution. So here I'm writing for the clusters of two molecules. And the reason why this quantity changes is because some of the GAG proteins in the cytoplasm arrive into the membrane and attach to it, and sometimes they detach. So they involve the number of proteins that are of the same size and the number of proteins that have one more size. I start with 2 and not with 1, and the probability of 1 does not appear here because it's known, because of the energies involved, that the clusters of 2 are needed in order to join the membrane. So our distribution starts from 2 and not from 1. That's really not important, but that's the thing. And there is also the possibility that in the membrane, there are other GAG proteins that are floating around, and they join this cluster growing. So if we put all of these processes into the equations, we can have a rate for the change of the size distribution. Of course, we have to write this for n equal to 2, 3, 4, and so on, 2,000 if you want. So conceptually, you have on the number of 2,000 ordinary differential equations for the full set of the probabilities. Okay, and I say conceptually, but it's really true because we cannot solve these equations analytically. So we have to solve it numerically. And in that, we need to write the whole damn 2,000 equations for the correct RNA channel and also for the wrong RNA channel. So we are solving on the order of 2,000, 4,000 ordinary differential equations. So the other thing that I want to say is that I use the symbol P for the distribution of sizes of actual infectious particle viral particles, the virions. And I choose the variable Q for the particles that are not really infectious. They are called virus-like particles. That's the name that people give to these empty shells. Empty in the sense that they cannot infect you, but because they don't bring any viral RNA with them. It's just that the structure is there, but it's empty of the viral RNA. And they're called virus-like particles, BLPs. So P is for the infectious, and Q is for the non-infectious one. Okay. I have to be patient because it takes some time to change. Okay. So if I do the analysis again, but for the virion assembly, the parameters change a little bit. So for instance, instead of the concentration of the RNA, I have to put the concentration of the viral RNA, and this psi is the ratio. So this psi is on the order of 10 to the minus 4, 10 to the minus 5. And I also have to consider the effect of specific binding to this RNA. This epsilon energy is extra to what I had before. So that's really not that much difficult. And we played with this on the order of 1 year and the beginning of the pandemics. So I also have to put an ordinary differential equation for the amount of GAG protein that is present in the cell. And that is controlled by the rate of protein synthesis. So if you have some rate delta, you have a new number of GAG proteins per minute or per second. This is the amount of protein that is stored globally in the virions. There are some copies in the clusters of size n, and so I have to take into account that number. And finally, you have these funny, strange combinations of probabilities, but what this amounts to is to the current at the end of the virion. When you assemble the full virion and it reaches size nm, then the cell ejects these things with a characteristic rate. The assembly of the cell ensures that the virion detaches and goes away from the cell. And we model this as a current that is, this will be like a discrete approximation of the derivative. So it's that sense, it's a current that's going away. If you solve this numerically, you can see what happens to the chemical potential that is related to the concentration of the GAG protein. And what it does is that it tunes itself to the energy associated with the binding. We didn't model this. It just happens when you solve the equation. What it does is that this concentration matches this, which says that the concentration of the GAG protein becomes equal to that of the dissociation constant. So the system tunes itself to that. Now let me show you some pictures. What is shown here is the size distribution. This is the probability of having a cluster of size 200. This is the probability of having a cluster of size 400. This is the probability of having a fully assembled virion of 1,000 particles. And this is a snapshot in time. What happens is that there is a shock that propagates to the right. So you see that the maximum here starts to travel and travel over there. What does it mean? This means that the average size of the cluster becomes larger and larger and larger over time. So that's what you expect when you are assembling the virion. Okay. If you wait for a long time, eventually you reach steady state. In that steady state, the probability is flat, except for this region, which is like a kind of a boundary layer. I'm showing you here two curves. The blue curve is for the virus-like particles, the VLPs, the empty viruses, the viruses that don't carry the actual infectious viral RNA. And there is a second curve that here is in red. That is the probability of finding a cluster that has the correct infectious RNA. So you can see that this model says you are never going to produce infectious viruses. this model doesn't predict any production of virions. In that sense, it's a failed model. So it always happens over here. What happens if instead of having the advective solution, which is this shock, the concentration is below the dissociation constant? Well, in that case, you don't have a front. What you have is a diffusive process that in equilibrium resembles this Boltzmann distribution. So the model performs well. The calculations agree with what you expect. It's just that this model cannot explain the selectivity. So there is something missing. What is that part that is missing? Well, I'm telling you this, but we were constantly struggling and changing the parameters and moving things around for about 1 year. And out of frustration, we remembered that we left out something in this model. And that something is dimerization. I'm going to explain this curve again. This is the probability of having a complete virion at time t. So if I wait some number of seconds, this is time divided by seconds, and this is about 1 day, I will have create a number of virions, of BLPs in blue. And the number of virions will be the curve flat at 0, essentially at this scale. So this model says, if you wait long enough, you will have a good chance of producing BLPs. You are creating your capsid, you are creating your box, and it's full of trash. It's full of RNA that is human, and that is not helpful for the virus. So this model is hopeless in that sense. It never gives you production of virions. And the thing that we need to do in order to create those virions is to include the dimerization. So just like we had a kinetic process with a rate that gives you how is the size of the cluster changing, we also have to include this. This is the dimerization initiation sequence that I told you before. This is a cartoon. And we say, well, the thing that makes the viral RNA different is that it has the possibility of creating a dimer. And we are assuming in here that this dimerization is very strong, that essentially this process is irreversible. So one way to think about it is this. You have a cluster in the membrane, has a large number of particles, has been growing for some time, but still kind of small. And then, over there, some other place, there is another cluster, has a similar size. In time, they explore the membrane, and after some time, they meet. And when they meet, they have some probability of fusing together. Sometimes, if they don't have this dimerization sequence, that will fall apart and go back, and they will continue their lives apart together. But sometimes the collision occurs, and both of them have viral RNA chains that are able to dimerize irreversibly. And if that happens, suddenly you created a much larger cluster. Okay, that's it. That's all that you need. One way to take into account this process, is to say, that there is another process that occurs here. And that says that there is some probability of a cluster of size m joining another of size n minus m. And then they collide, they create a cluster of size n, increasing your probability of having a cluster of that given size. Also, you have to take into account the opposite process, that you have a cluster of size n that has collided with something else and has become something that is no more one of these guys. And this is from the theory of living polymers. When you do that, then you obtain what you want. Now, under the same conditions, the red curve that represents the number of virions produced per unit time suddenly becomes larger than the number of VLPs. And what happens is that this occurs for a small amount of time. This selectivity is pronounced only for a small number of hours. The small number here represents on the order between 24 and 33 hours. Over that period of time, the cell is producing mostly virions. It's also producing VLPs, But the number of virions that is produced is much larger than the other one. Because they have this advantage. These clusters that are small collide, become a much larger cluster, and that promotes them to much farther away in the race. So you are giving the probability of reaching the end a big jump because of that. But eventually, these things will reach equilibrium conditions, steady-state equilibriums, and you will lose selectivity. Well, we spoke to the experimentalists that say, well, that can happen. Initially, they were a little bit concerned because they said, well, these are immortal lines. So that means that the cell doesn't die, and so you will eventually find the equilibrium conditions. But the person that actually does the experiment says, well, the cells do die because when they are infected, the immune system of each cell detects that the cell is ill and eventually kills itself. So the cells don't die, don't live together. And they typically measure the selectivity only at 24 hours after the infection. And that's the data that the experimentalists measure. And we're saying, well, for that range of times, we will observe this. But we also make a specific prediction. We said, if you wait more, the selectivity will be reduced. And it's hard to find in the literature if that happens or not. One of my co-authors, Julia Rosina, happens to know the person that makes these kind of experiments. And she asked, well, have you ever tried to measure the selectivity at longer times? And she said, no, we never do that. Why not? Because it's smaller. So we have indirect information that says that this probably could be okay. So let me go back. So we were speaking about the possibility of making some predictions that can be tested against the experiment. One of the things that you can do is to mess with the probability that the dimerization will occur. So say that the probability of dimerization is very large, on the order of 0.9 or so. Then you measure the amount of virions compared to the number of total number of particles that are created. And if you put that number here, you get selectivities on the order of the 90% or so. If you start to reduce the probability of getting fused on collision, and you make these probabilities more and more and smaller, eventually you reach very, very small selectivities. So this is something that we say. If you mess with the dimerization sequence and make more improbable that dimerization will occur, you will lose selectivity very fast. That can be done. Can be, actually has been done experimentally, and that's what they observe. If they sequester the dimerization sequence, the selectivity is lost. So that coincides with something that is observed. So the model survives this. There are some good news and bad news. The bad news is there is a lot of parameters in the model. So that means that we have to rely on a lot of experimental information that has larger bars. Now, the good news is that there are a lot of parameters in the model. So that means that there are many ways that you can convince an experimental friend to make some change to the protein of the RNA and see if they can tune the parameter in a range that you can achieve. So all of these parameters, we have run the model to figure out what would happen. So for instance, one of the parameters of the model has to do with the value of the GAG-GAG interactions. If they're very strong, then the GAG concentration required to initiate assembly is small because they interact very strongly. If that interaction is weakened, then the concentration needed to initiate assembly becomes much larger. So this means high interactions, this means low interactions. And the specific prediction is if you reduce the interactions, you will lose selectivity too very quickly. That can be done because we know from simulation and from experiment the parts of the protein that interact each other when they act together in the membrane. So if you create mutants that decrease the size of the interaction energy between these parts of the protein, you would expect to see a big loss in selectivity. This hasn't been done, but could be done. In fact, we are talking with the experimental friends of Julia to see if that can happen. Another thing that is characteristic of this virus is that it's very, very slow producing its own GAG proteins. The synthesis rate of the GAG protein is very small. And that seems like it's funny. Let's see it this way. You're trying to grow a house, and you want to make it very, very selectively. So you need some bricks. And what I'm saying is that the cell creates bricks very, very slowly. The GAG proteins needed to create the house are produced at a very, very slow rate. And why is that? If you have to win the race, wouldn't you want to make it as fast as possible? And the model says no. What you need is to create these bricks very slowly. What is that? What is here is a series of plots in different colors that give you the fraction of viral RNA in total. So that's like my psi parameter, the small number of copies of viral RNA compared to the total RNA that is there. So this is a whole lot of RNA, and this means a very, very poor amount of viral RNA. And what I have here is curves that are created in the model for fast production of the GAG protein or for the slow production of the GAG protein. And what happens is that these curves are shifting to the left. So that means that if you make progressively slower the creation of the GAG protein, you can reach high levels of selectivity at very small amounts of viral RNA. So in that sense, this explains how the HIV works. The HIV can get rid of the necessity of having a huge amount of viral RNAs if it makes the production of GAG proteins very slow. If that happens, you can reach high selectivities with very small number of copies of your viral RNA. You don't have to waste it. Okay, how am I doing with time? Okay, so essentially what's going on here is another list. Well, I'm going to do it. The other thing that you can do is, instead of changing the protein, changing the viral RNA sequence. You can make mutations in these sites in order to mess with the total energy of binding the RNA by the GAG protein. So this is another possibility. You can change that interaction. And this is that energy. So if you can imagine that maybe the interaction energy is about here for a normal virus, and if you reduce it, the prediction of the model is that it should go down very, very fast. So these predictions can be used to try to convince our experimental friends to actually do the investment to create all these mutants and changes in the viral mechanism and try to test it in vitro first. So what we like about this is that so far, this is the first model that allows you to predict that you can create high selectivity at very small amount of viral RNA without having to pay a lot of energy by specificity. In that sense, it matches what the experimentalists see. And it matches that over a small period of time, you will get a lot of viral production. It will not last forever. If you wait long enough, that will be steady state, and in steady state, you will recover the low thermodynamic limit. So that's another specific prediction that we do. And we are at that stage. We are trying to convince our experimental friends to actually perform these things. And also, you can think that this is a strategy to create assemblies. Well, maybe the virus does this. Maybe it doesn't. But engineers could use it. If for some reason you would like to pack things that have a small amount of something, one way to ensure that is that you enhance the growth by this kind of mechanism. So even if the virus doesn't do it, it will be possible to synthesize systems that take advantage of this mechanism. So there is a win-win situation. If the virus does it, we'll be very, very happy. And if it doesn't, then it will be a new mechanism to enhance the production beyond the limits of the thermodynamic limit by a kinetic mechanism. And with that, I will I will stop, and I'll try to keep my conclusions here. So what we have provided is, for the first time, a simple phenomenological model that is consistent with the biology of the HIV virus, and explain how is it that it gets away of not having a tremendous thermodynamic advantage. I would say it has a kinetic advantage that is given by the specific dimerization mechanism that the RNA in this virus allows to have. And it's not 2 copies, it's 2 copies exactly, not 3, 4, 5, whatever. It's just 2 because the chains are complementary in that part. The parameters are obtained from known experimental data, and they can be tuned if you create synthetic versions of the RNA or the proteins and mutate the regions that are providing you the parameters of interaction that we have modeled. And we believe that we have some interesting predictions that can be tested experimentally, and we hope that in future we will attract that interest and the theory can be tested empirically. And with that, I thank you very much for your attention.
Questions? All right. Very nice talk. Oh, you.
I just have a very brief question, Orlando. The scale of the probability in the first plot that you put is like 10 to the minus 8 or something like that, and the other one I didn't see it.
Yeah, these probabilities are very small because they are associated with a single site.
And that
single site.
That is what is expected because 10 to the minus 8 is, right. Well, it depends, but so it's almost 0, right?
The wheel works.
Yeah.
Each site is one of those yellow spheres. And so the number of spheres can be under of several thousands or so. And that probability also depends on the specific number of the parameters that you use. And the smaller the synthesis rate gets, the probability increases. So it's an example, but you can increase it by the number, you can scale it. Yes.
All right. Well, we thank to Orlando for this enlightening present. So now we have our last talk this afternoon, and then we have the winner of the poster contest. All right, the winners. So we have Pavel from UNACH, right? From the Autonomous University of Chiapas.
Okay, thank you very much for the invitation to this wonderful meeting, BioPhys 2022. I would like to thank especially to Luis Olivares for this kind invitation and also for all the committee, the organized committee of this conference. Well, as the Dr. Eduardo, mentioned, I come from Autonomous University of Chiapas, which is at the southeast of Mexico. Here in this photograph, I show the buildings where the Faculty of Physical Science and Mathematics are, and there for the undergraduate and also for graduate students, we have programs, Master in Physical Science and also in Mathematical Science, and soon a PhD program. So you are welcome. Also for all professors, you are welcome. The buildings are far away from the city center of Tuxtla Gutiérrez, so you can focus in your research. Okay, before to get in the talk, I would like to mention briefly a preamble of the origin of the problem that I'm going to talk. Let me show you here. Well, also motivated by a biological physics problem, I was interested some time ago to study Brownian motion in curved surfaces, especially to study how proteins, integral proteins through the membrane, are moving through a cell membrane, for instance. So we were producing this result for the mean square displacement in terms of geometrical quantities of the membrane. So just to mention, let me, if I can, when we study a membrane from this perspective, basically what we think of the membrane is a surface, a mathematical surface, like this back. So we can consider this surface as basically, in the case of the membrane, produced by the hydrophobic effect, by these amphiphilic molecules. So one can think, and very rough approximation, that you can characterize all the Brownian motion of a particle which is constrained to move in the surface in terms of the curvatures. So basically a surface has two curvatures. One is called Gaussian curvature. The other one is called mean curvature. Here in this equation, this RG is the Gaussian curvature. For instance, this cup, basically the cylinder doesn't have a Gaussian curvature, just have a mean curvature. So, but the sphere has a Gaussian curvature. No? So depending on the surface, well, there may be a surface that has both curvatures. So, but interestingly here, this mean square displacement, which is associated to the geodesic displacement, I mean this smallest distance between 2 points, depends just on the Gaussian curvature, okay? So this is analysis just for a small time. And it was surprising that in 2017, these people from North Carolina, they performed these experiments where with drops, oil drops, and they put nanoparticles in the interface of the oil drops, and they measure with single tracking experiments the Brownian motion of these nanoparticles in the interface, and they show basically this equation. So it was a surprise because this was very ideal situation, but they got that. And actually, we also have a similar result for the active particle motion. No? This, so how I got, how I go to polymers, which is the central part in this talk, is because it is known theoretically that Gaussian polymers that they were talking yesterday by Dr. José Luis Vélez, if you study the geometry of the polymer, the conformational geometry, they look very similar to the Brownian path, the Brownian motion path. So actually, the same equations can be used to describe the conformation of the Gaussian polymer. But after a while, Montesquieu and Muthukumar also, they wanted to study these polymers in surfaces for different reasons. I mean, surfaces can be used as a model to constrain the polymer for different situations. So they were studying theoretically in this sphere, cylinder, cone, and also in torus. And actually, using our theory for the Brownian motion in curved surface, we can also study this quantity, which is called the mean square end-to-end distance, which is related to the radius of gyration of the polymer in terms of the curvature. Because of this quantity is a quantity which is embedded in the space, all the dependence is just extrinsic curvature, this mean curvature, which maybe people that know a little bit of differential geometry, mean curvature is the sum of the principal curvatures. A principal curvature you can mention if you take the surface, put the biggest circle. So the inverse of the radius of this circle is one of the principal curvatures. So this is the reason why I was interested in the polymers, because of this connection. But polymers are flexible, no? Like this. So if you try to bend a polymer, no? Like you are describing here the polymer as a curve in space, so there is some rigidity that is opposed to the bending, okay? So the question theoretically was, what are the stochastic motion that describe the polymer, no? As was the Gaussian polymer described as a Brownian path. So it was just a theoretical question. Let me see, okay. So, well, what are we going to talk in this, what are we going to be discussing in this talk is basically these two papers that we were published in Physical Review A and also Frontiers in Physics. But let me go to in the details. So the outline of my talk is going to be introduction and motivation, more in the context of the biophysics. I will show the methods, which is a new method that we developed that we call stochastic curvature method for semi-flexible polymers that we are going to apply for DNA, basically. Yeah, and the results, which is the main title of the talk, a universal conformation, transition between order and disorder states for a semi-flexible polymer. So, well, the motivation are the study of macromolecules. Macromolecules play a central role in different branches of science, like chemical technology, and especially in the context of this conference, biology. Many systems comprise polymers like petroleum, liquid crystals, etc. And we are focusing in linear polymers. And these linear polymers can be flexible, like spaghetti, or also can be rigid, like a rod, okay? So especially, one of the interests is to study the conformations of biological molecules, because, as you know, I don't have to mention, like proteins, DNA molecules, RNA, etc., can be considered as a polymers in a first approximation. So one of the motivations to study the conformations of these semi-flexible polymers is the problem of DNA and RNA packaging in viruses. And we saw the talk by Jaime, very interesting, where they talk us about all the physical information about the virus, behind the virus. So I don't have to be more extensive in this. But basically, we are idealized the problem, considering that the polymer is confined in some region. So we wanted to understand what are the conformational states of the semi-flexible polymer that is representing DNA or RNA inside the virus. Actually, we are focusing in the case of DNA because this is the one that comprises as a linear polymer. No? The other is a little bit more complicated. So there are some facts for viral DNA. Basically, it is known by different spectroscopy X-ray experiments that viral DNA is folded into a coil-like structure, and there were some models since 20 years ago that they can describe this conformation, this like a toroid conformation that was mentioned also for the Professor Jaime. And also in this paper, Spakowicz and Wang, they study the play, how the twist of the polymer may play. So they mention this, they show these conformations. And there are other information of the viral DNA. For instance, as was mentioned also, the pressure inside the capside is around 10 to 40 atmosphere. And this play an important role when the virus infects the people. or the other organisms. Also, it is known that there are electrostatic interaction between DNA itself, okay? So actually there is a very nice recent paper by these people, Efremov, Ladislav Hoban, and Jie Jan, and they study what is the relation between the nucleosides and its effects on nucleosome stability in living cells. Actually, they are studying on a statistical physics model, which is on the basis that it is called polymer field theory. What is the relation between the structure of the chromatin and the nucleosides? But one interesting thing is that they analyze their model in an example, which is the viral DNA, in order to see if all the things are correct. So these are the results. I mean, of course, they are including, is a very, I would say, complete analysis of what is happening with the DNA inside of the capside, because they include the ions, they include the electrostatic repulsion between the DNA with itself, and they are allowed to understand, for instance, the free energy per DNA, the mean capside potential, and other quantities like the pressure inside the capside. But I am interested basically in these two quantities, especially in this root mean square displacement, which is the quantity that I can compare with my formalims. Basically, they mention this WLC, which is the warm light chain model, which is a model that basically considers the polymer as a cure, no? Like this connector. And, but actually, if you see in this image, the capside radius increase here, and they put the WLC model to the limit, okay? But I will argue here that the WLC model still reproduces this curve, these oscillation curves, which is a feature that is characterized this coil-like structure of the DNA. Also, this is in relation with these oscillations of the correlation between the tangent vectors of the DNA. Basically, tangent vector, it refers that if you take this ending of the polymer and this other ending, and so what is the direction, okay, in the ending is going to be a tangent in this final point and the other tangent. So you can compute the correlation between these 2 points, and they oscillate, and this is a characteristic of the, I mean, they give you information of how the conformation of the polymer is inside of the capside. Okay, so we are going to be simpler because we are not going to consider the electrostatic repulsion, the DNA, and also we are not going to consider the ions in some sense, but let's see, because we wanted to see what is the power of this model, the WLC model, no? What can give us in general. No? So we are going to focus in this quantity, which is the mean square end-to-end distance. As I mentioned, basically, if you have the polymer, you take the distance between this ending point to this ending point, this linear distance, and this is this quantity, okay? And then you take the square, and you take an average over all the possible conformations of the polymer, all statistical conformations. So the point here is, what is the statistical physics theory that describes the distribution of these conformations? So let me hear, well, this, let me say something more. This quantity is also related to the radius of gyration, which also it can be measured using spectroscopy experiments, like the one that was performed more than a century, well, half century ago by Kratky-Porot. They performed X-ray experiments on molecular chains, basically vinyl bromide in cellulose nitrate solution, and they got this equation experimentally, just from the experiment, and they adjust this quantity, which now we are going to call a persistent length, and already this equation can give us information of the elasticity of the polymer. If we take the limit, for instance, when we take this capital L, is basically the size of this polymer, and when we take this L much more bigger to this LP, or vice versa, LP much more smaller than this capital L, so the mean square displacement is linear in this length of the total length of the polymer, and the behavior of the chain is basically the trajectory of a Brownian path, as you can see. But if you increase the value of the persistent length, the polymer makes more flexible and then a very rigid rod. So the people like Hermans and Ullmann in 1952, they, on the basis of Brownian motion theory, they extend basically the Brownian motion theory to study the conformation of the polymer, and they got to this equation. Basically, this equation describes the probability density function, okay, to find the polymer with size S, okay, with size S, and with endings XY and X0 and Y0, with the directions theta and theta 0. Basically, this is the equation that we were looking for to describe the distribution, basically, of the endings of the polymer. So this equation has information of the different conformations of the polymer in the environment. And they show that this equation reproduces correctly the Kratky-Porot equation with this parameter, which is the inverse of the persistent length, but they didn't justify, just extend as a Brownian motion theory, because they were thinking that this is the manner how the polymer is conformate. So it is an heuristic model at this point, but they reproduce this correctly. After some time, Saito, Takahashi, Yunoku, they got, they say, well, but the polymers are elastic, so we should be able to describe the conformations under the basis an elastic model. If you can see, this is an elastic, this is the basic elastic model to describe a single chain. And using actually a path integral, Feynman path integral formalisms, they were able also to reproduce the Kratky-Porot formula and also explain what does mean this persistent length. Persistent length, basically, they found that is proportional to the rigid of the polymer. And rigid has information of the manner how the single molecules, nucleotides, and all the structure of the molecule of the polymer are interacting. So, they found that the persistent length is proportional to this rigid and rigid coefficient, and also inversely proportional to the temperature. Okay, and so if you have a bigger temperature, basically you will have LP smaller, and then the polymer will behave as a Brownian path trajectory. Okay, so, and let me mention that it was one of the triumphs of this model, the WLC model, which is the same as Saito's proposal, is this experiment by Bustamante, Marcos, Siggy, and Smith in 1994, because they, using optical tweezers, they got, they took the endings of lambda DNA, and they impose a force, and they study the extension of the lambda DNA against the force, okay? So they got this curve, which with these squares, and this dashed, the other curve is the one associated to the Gaussian, which is described as a Brownian path. But this is the good one, which actually, if you can see, there is a black line under the squares, is basically this formula that you can get analytically or estimate analytically using the WLC model, okay? So, at this point, one can say that this is a good model. This is a good model in some limit, well, when you are ignoring somehow the secondary and the tertiary structure of the molecule, but it's a good model to describe the conformations of this DNA. So, well, let me see. Okay, so our perspective is to consider the model as a curve, continuous curve. So we have to go and take some tools from differential geometry, especially in this case, differential geometry associated to curves. It is known that every curve satisfies these three equations, which are called Frenet-Serret equations, which has information, structural information, of the curvature of the curve and the torsion of the curve. For instance, if you have a polymer, which is, it is on the plane, okay? Well, I am trying to put, this polymer in the plane, okay? Suppose that it is in plane. So it doesn't have any torsion, okay? Torsion will be 0. But if you take the polymer out of the plane, so there's going to be a torsion. The DNA molecule in the space will have a torsion. This double helix will have a torsion. So, but every curve satisfies these equations. There are some geometrical quantities like the tangent, which is basically vector, which is tangent in each point of the curve, the normal vector, which is perpendicular, and the b-normal, which is perpendicular to the plane built by these two vectors, tangent and normal. So this is our model in terms of geometrical quantities. This is the same elastic energy that used Saito, Takahashi, Yunoku, and that one that was proved by Carlos Bustamante in these experiments. And from the theoretical physics perspective, it's a very nice model because it's a geometrical model, and you can consider this as a functional in terms of this parametrization, and you can take, I would say, the minimal value problem, and there are some nice properties like rigid translations and rotations. Of course, I mean, the elastic energy that my polymer have doesn't change if I just translate or if I just rotate, okay? There are some invariants that you can put mathematically, and then, and also, is invariant on the reparametrizations of the curve. I mean, it doesn't depend how I measure the curve. So these are features that has, very nice features that has this model. But unfortunately, this is not like an oscillator model. Like, I mean, there is this joke that in physics, everything is an oscillator, but it is not. Actually, it's non-linear in this dynamical variable that represents the points of the polymer or the curve. Okay, so it's a non-linear action or energy. Now, we wanted to study the different conformations of this model, but when we have thermal fluctuations. So there are different perspectives that the people have used to study the thermal fluctuations. For instance, they can go directly to the configuration space and try to count all the possible conformations and try to do your statistical physics to study the mean values associated, like the mean square displacement, etc. But actually, one can go also to the tangent, to the space of tangents, because this is also a space that is over there, and also to the space of curvatures. And actually, when you pose the problem of statistical physics, this is the thing that we wanted to study. This is the probability distribution of these positions of the polymer. But it is a very complicated to obtain, for instance, the mean quantities using this probability distribution due to the fact that this action is nonlinear in this quantity R. It is encode the information of R because this S, which is the arc length of the polymer, depends on R. I mean, it is high here, and also kappa, which is the curvature, depends nonlinear on R. And so it's basically, it's not possible to compute the expectation values from this perspective. And the idea, actually, Saito, Takahashi, Yunoku, how they got this Katki-Porot equation is that they make the observation that kappa square is actually the first derivative of the tangent vector. So you can ignore a lot of information if you integrate just in the space of tangents. So if you have quantities that depends just on the tangents, so you can compute quantities because this is already a quadratic action in the tangent. So this is the manner how they were obtaining these results. So we go further and we say, okay, we can pose stochastic Frenet equations because we see that every curve satisfies these Frenet equations, but we are going to pose this stochastic Frenet where the curvature now we promote to a random variable, which has the information of the thermal fluctuations. So if you are interested to study the remaining geometrical quantities, like the normal stochastic vector, the binormal, or the torsion, you can, if you know the manner how this quantity is distributed, so you can compute it from here. The second postulate is that what is the distribution of this random variable, the curvature? Well, is this one, okay? Based on the WLC Hamiltonian, okay? Which is the same that we were studying. So, but now, it depends, I mean, this functional distribution, it is simpler because now we don't have any derivative here. So actually, as was mentioned yesterday, if you compute the correlation between the mean, the curvature, we are going to have a delta correlation or white noise theorem, a kind of fluctuation dissipation like theorem. And then it is simpler, no? We, for instance, in the case of a polymer which is confined to the plane, we are able to show that the equations that appear are very similar to that one in Langevin equation for active particle. So this is the mathematical question that we were asking at the beginning. Actually, the paths that the polymer, the semi-flexible polymer describes are those that the active particle perform in the space or in the surface. Okay, and we can go further and put the polymer on a curved surface, okay? Ahora, now we are going to have a surface, and we wanted to constrain the polymer on the surface, and we can extend all the formalins to write down now this stochastic Frenet equation that capture the information of the surface through these red coefficients. And we can get mathematically a generalized Hermann-Ullmann equation, but now for the distribution of the conformations of the polymer, but now that is constrained to a surface in terms of the geometrical quantities like the metric, which basically if you have the metric, you can determine, for instance, the Gaussian curvature and all the intrinsic geometry. Okay, and we can, for instance, choose different values for the metric, and for instance, for the case of the plane, these are the quantities that we need to choose, and these ones reduce to the Hermann-Ullmann equation in the plane, or the space if you want. Now you can treat this equation in a multipolar decomposition and find an infinite tower of hierarchy of differential equations that will describe all the information of the polymer. So it is difficult to deal with all this, but we can see that the coefficients in these tensors is increasing in value. That means that there are some regimes where the bigger tensors will not contribute too much to the conformation of the polymer. For instance, this PA, it says how directed is the polymer. This is relation to how the tangent ending directions are correlated, etc. Okay, well, this is our theory. Now, let's go to applications. Well, just a last implication is this very nice equation that we found, which is the telegrapher equation, a modified telegrapher equation, because it involves this tensor that has information of the correlation of the tangents, of the ending tangents. Okay, but we can see something here. Yeah, from the equation, we can say already physical information, because you can see that if we ignore this correlation, we can argue because of these coefficients as I was mentioned, and we also put the persistent length goes to 0, or otherwise the temperature, a bigger temperature, we can observe that this equation reduces to the diffusion equation in the surface, or the diffusion equation, which is the equation that describes the conformation of a Gaussian polymer, okay? Basically, a Gaussian polymer that it behaves like this conformation, okay? So this has the information of the elasticity of the polymer. Now we go to the results. This is a universal conformational transition between order and disorder state for the semi-flexible polymer. How much time do I have? How many? 10. Ah, okay. Well, more or less. Okay, Spakowi wants in 2003, in this perspective, to study the properties of this model, the warm light chain model. They study a polymer confined to a spherical surface, okay? So they found this behavior for the mean square end-to-end displacement. Basically, they took a persistent length less than this quantity, which is proportional to the radius of the sphere, and they found that the mean square end-to-end displacement has a ballistic behavior, okay? And then a linear behavior, and then a saturation to a plateau, okay? So basically, this is the behavior that a Brownian particle has in a sphere, basically. But when you go to a persistent length, which is bigger than the radius, so it appears these oscillations in the mean square end-to-end displacement, which you explain as this coil-like structure that the DNA is having, but now here in the sphere. So we can think as the sphere as a model of the cap safe, okay? And you can think that the polymer, the DNA polymer, because of the DNA electrostatic interaction, this interaction, this repulsion, will put the whole polymer in the surface of the sphere. So it's somehow a manner to model this situation. So these are images of a simulation that we performed in a collaboration with my collaborator, Johnny Ramirez, from the WAP. And we reproduce correctly the shapes. Actually, they perform analytically all the results. And we show that using our formalisms with these stochastic Frenet equations, we reproduce correctly the expected behavior. Okay. So actually, we can do also analytically in a very few steps. I mean, they made some kind long calculations, and actually with our modified telegrapher equation, we can compute this mean square end-to-end displacement, substituting here because the quantity exactly, actually, this quantity cancels because this Q is traceless, is some, and then we got this equation that describes this conformational transition. Now we can ask what is the origin of this conformational transition, and actually when we arrived to this problem, we wanted to study the semi-flexible polymer on surface, how the geometrical properties like the curvatures are affecting the conformations of a general surface, okay? But we arrived to the surprise that even the case of a polymer inside of a square, very academic problem, was not solved. So we solved this problem using our formalisms. And our surprise is that under certain conditions, the same formula, mathematical formula, as the Spakowy-Wantz formula, we found. So we found basically a conformational transition also in the square for a polymer which is inside of the square. Okay. We go further. Well, this is the same, the semi-flexible polymer confined to the flat box. And here we show some image of the manner how the polymer is inside of this square, and how you can see that the polymer is soft in the way that the persistent length is increasing. Okay. Now here the polymer is soft, is softer than here, that it looks like a Brownian path motion. Now we can go to simulate a situation more, well, not realistic really, because actually the capsides are not spheres, but approximately you can think that they are a sphere, and you put your polymer here inside of the cube and the ball, and you also find a similar expressions under certain limits between the length of the polymer and the persistent length. So they show also this transition, conformational transition, between this disordered state, which is the Brownian path, to the ordered state that will describe this more soft path or soft trajectory of the polymer. And also, we can, well, these are the plots. Here now also are the mean square end-to-end displacement for the case of the cube. And we can see that for these values of the persistent length, the conformation of the polymer looks very, very similar to a Brownian motion. But now when we increase the persistent length, the polymer occupies almost all the region inside the cube, and the trajectory is softer a little bit. No? So this softness that has the conformation of the polymer is basically described or featured by these oscillations that has the mean square end-to-end displacement, and also coincide qualitatively with the one that found by these colleagues Efremov using this polymer field theory that we showed at the beginning. So basically, this is showing that the conformation inside of the capsule, this coil-like conformation, is basically governed by the elastic properties of the molecule, of the DNA molecule. But we go further, and we study also conformations on a toroid, because we have this perspective that geometry is more than academic or a play thing, because as you can see in all the conference along this event, this biophysics event, the biological systems from the physical perspective are very complicated. So study, for instance, the ions that are interacting with the DNA inside, controlling exactly how is precisely particle by particle, it is very difficult task. So we can use surface to constrain the polymer, to say, okay, I don't want it that the polymer is allowed in that region. So we can use the formalins to restrict the polymer to some regions in the space, and this surface may model some effect, for instance, the electrostatic effect that the ions inside of the capsate are performing on the conformation of the polymer, or other effects that are affecting the polymer. So we study also the phase, the transition. Now we can see here a persistent length LP 0.1 over the radius of the torus, and this is a very small persistent length, and we can see also a very accidental conformation, that this is reflecting the fact that it's a Brownian path. But if we increase the value of the persistent length, also we got a softness of the conformational behavior of the polymer. Actually, now, because of the toroidal surface, is a little bit more complicated than the sphere, etc. We have regions which they have negative Gaussian curvature, a positive Gaussian curvature. So we have two parameters, basically, the inner radius of this circle and also the external radius of the whole shape. And then we can plot a diagram, phase diagram, solving these stochastic Frenet equations. Here in this axis, we show the persistent length against the ratio between the radius. So the black dot points represents the behaviors like I would say monotonic increasing behavior in the mean square end-to-end displacement. Basically, this is a typical conformation for a polymer which is in this region, okay? But if we cross this interface, so we are going to arrive to a region where the conformation of the polymer is soft. But we can go further. Here is just to persistent length to 1, but we can go to persistent length of 100, and we can see that the polymer is still more soft. And if you go just changing a little bit the radius, we can concentrate the polymer inside of this circle of the torus. So we can use the torus in order to understand or model, I would say, for instance, these toroidal behaviors of the polymer inside the capsate. Okay. Also, we can use our formalins to study the conformational, this this transition, this conformational transition, on a cylinder. And this more or less mimics the situation that the genome performed, for instance, in the histones, the octamer histones in the chromatin structure. So maybe one can do some explanation of this circular situation using also the elasticity perspective. Okay, I am now finished. We can do general, actually, on a general surface, and we have a good results that this is under certain conditions of the size of the polymer and the persistent length, also related to the first eigenvalue of the Laplace-Beltrami, that this is a general equation to describe the conformational polymer described through the mean square end-to-end displacement in a general curve space, which is compact, which means that the polymer is confined in a region. So there is a good expectation that this transition occurs for every semi-flexible polymer in a compact space. Thank you very much, and I would like to acknowledge to my collaborator, Dr. Johnny Ramírez from UAP, from Benemérita Universidad Autónoma de Puebla, and also to all the financial supports, my university, the CONCITEP from UAP, and also our national financial support, CONACYT, and thank you very much for your attention.
We have time for questions. Yes.
In the expression for the Hamiltonian, you have the curvature of the polymer, and the curvature of the surface is encoded in the inverse of the curvature G. That's the curvature of the surface, and the Christoffel symbols. Yeah. So what I'm thinking is this: in the case where you have the polymer confined to the region of negative curvature in the inner part of the torus, it seems like it's interacting. It's being driven to the negative curvature of the surface. Not that curvature, the curvature of the surface of the torus. Is there any alternative representation when you can kind of show how is the curvature of the surface interacting as a kind of a field or something dragging the polymer inside? I've seen this type of argument for the topological defects in liquid crystals, where the free energy of the liquid crystal involves the curvature of the surface, and then you can see the topological defect dragged into the regions of negative or positive curvature.
Okay. We have not analyzed from that perspective, but I think it would be good expectations to do that. And if I understand your point, is that basically we have to study how the Gaussian curvature in the region of the negative region change under this parameter. Okay, and maybe there is a good expectation why the polymer collapse into this region. Okay, well, I don't know exactly the answer, but I suspect that it has to do with the first eigenvalue, or the eigenvalues in general, of the Laplace-Beltrami, because there are estimations of the eigenvalues in terms of the curvature properties of the surface. So maybe in the negative region, it happened that. But the other thing that also we should mention is that this expression is calculated in the region, in certain region, okay, where persistent length is not very large. Actually, we suspect, well, we are trying to see how much it is valid, this equation, okay? Because this situation here is for a persistent length very, very large. So when you have a persistent length very large, the stochastic behavior is basically neglecting, okay, vanish in the manner how this increase the persistent length, so the stochastic behavior of the conformation of the polymer is start to vanish. So, but the equations that appear are basically the geodesic equation of the geodesic equation on the surface. So the geodesic equation also has information of the curvature. So maybe in that way we can argue the situation. Thank you very much for your question.
Any other question? No? Okay, well, thank you very much, Pavel.
Thank you very much
for this interesting. And we would like to give you a small present from the conference. Thank you very much. Okay, everyone, so ya pasamos ahora a la parte de, bueno, hemos terminado prácticamente la conferencia, entonces pasamos a la parte de la premiación de los pósters, ¿verdad? Con Eduardo y Carlos pasamos acá. Entonces, ya no vamos a hablar en inglés, así que ya vamos a hablar en español. Ok, bueno. Carlos, vente para acá. ¿Cómo está esto? Para acá. Sí. Si quieres empezar, o Eduardo, no sé. Bueno, ¿quiere que diga Eduardo?
Bueno, pues a la última etapa, muchas gracias por permanecer, por quedarse. La verdad es que espero que hayan disfrutado el congreso y que haya sido muy enriquecedor para todos. Y bueno, pues ahora tenemos un pequeño regalo para aquellos pósters que consideramos y bueno, nuestros investigadores nos recomendaron como para premiarlos, ¿no? Entonces tenemos 3 lugares. Tenemos ya los nombres de los premiados. Sí, entonces yo digo el tercer lugar, tenemos un tercer lugar para…
Tercer lugar para Marco Antonio Ramírez Martínez.
Tan lejos. Muchas felicidades, Marco, y a seguir adelante con éxitos y pues a seguir trabajando, que así es esta historia, ¿no? Felicidades. ¿Quieres saludar aquí también a ellos?
Muchas felicidades.
Gracias.
Muy bien, ahora tenemos un segundo lugar, ¿dónde estamos? Sería Jessica Pereda Méndez con el trabajo sobre redes. Está por allí.
Muchas felicidades, Jessy, que sigan los éxitos. Felicidades y a seguir trabajando. Muy bien.
Y bueno, pues les tengo la mala noticia que no hubo primer lugar. No es cierto. Bueno, el primer lugar es un disco externo de 4 teras, así es que vale la pena el primer lugar. Y este es para Juan Manuel Molina.
Juan Manuel Molina, ¿está aquí? Perfecto, felicidades.
Felicidades.
Muchas felicidades. Que sigan los éxitos y a seguir trabajando. Perfecto, pues nada más creo que a lo mejor decir algunas palabras finales. Para otorgar estos premios, pues consultamos con profesores que también estuvieron en la conferencia, ¿no? Entonces ellos nos dieron su opinión, estuvieron ayer revisando los pósters. Agradecemos también a los profesores y al comité que participó en esta evaluación. Muchísimas gracias por su apoyo. Y pues creo que ya vamos a la parte de cerrar el evento, ¿verdad? Y pues de mi parte yo voy a decir algunas palabras y luego le cedo la palabra a mis compañeros. Primero que nada, agradecerles yo en particular, a ellos dos en particular, a Eduardo Jardón de la UAM Lerma por todo el esfuerzo y a Carlos Muñoz del Instituto de Ciencias Físicas de la UNAM. Un aplauso también para ellos, por favor. Muchísimas gracias por ese esfuerzo. A continuación, a todos ustedes que estuvieron el día de hoy y ayer, a todos los colegas que participaron como speakers, a todas las autoridades administrativas de la Casa del Tiempo de la UAM, muchísimas gracias, maestra, por allá está, muchísimas gracias, le agradecemos, maestra, un aplauso para ustedes también, muchas gracias. Muchísimas gracias por su paciencia y bueno, sobre todo a ustedes, gracias a ustedes que participan y hacen esta conferencia, y yo nada más cerraría diciéndoles que corran la voz, vamos a seguir teniendo conferencias de esto en 2 años más, tenemos el BioPhys 2024, pero todo el tiempo estamos haciendo actividades en las distintas instituciones. Le voy a dejar la palabra a mis compañeros para que digan algo.
Muchas gracias. Bien, muchas gracias, Luis. Bueno, pues yo nada más agradecer que hayan aceptado la invitación a participar a los profesores investigadores y en particular mi interés en este tipo de actividades es sobre todo para los jóvenes, que se den cuenta que bueno, aquí hay trabajo en México, trabajo destacado y que está en el área de la física biológica o biofísica, que se motiven y tratar de hacer una comunidad pues, entre ustedes los jóvenes que vienen atrás, nos vienen empujando y que nos ayuden a que esta actividad sea constante y vaya creciendo y el día de mañana pues ustedes estén aquí, ¿no? Representando a la biofísica de México. Entonces, en lo personal mi intención en estar en esto es por la gente que viene atrás, ¿no? Que esto siga.
Sí, bueno, pues finalmente tener una red de colaboración, una red de apoyo mutuo y bueno, pues los intercambios que podamos hacer en el futuro, pues se van a germinar después de estos eventos, ¿no? Entonces, pues muchas gracias por su asistencia y pues nos vemos en 2 años. Gracias.